start my section. So we all know that the coronavirus outbreak has occurred in December 2019, especially numerous patients from Wuhan city, China were the first to exhibit conditions which were very similar to flu. And led the acknowledged this growth of virus and came to a conclusion that this virus is very different from SARS virus, and hence it is being regarded as the COVID-19 or SARS-CoV-2. And from that point of facing due to this outburst of the coronavirus that was shown in television, social media, or you take newspapers, and the discussion between the families and friends, as well as other sources, it resulted in some sort of emotional drift as well as panic situation among the population. So although people started to read about that there is a lack of proper kits and medical safety guards, as well as lack of proper beds, so this started creating anxiety and panicness, and later they started to think what will happen next, when the appropriate circumstance will return, so when the circumstance will come to normal situation. So these all things caused psychological trauma among the population. The main people who faced this kind of psychological stress or trauma are the healthcare workers as well as medical professionals, doctors, who are daily facing this kind of mental illness, because they are attending all the patients with COVID-19 cases, and they are seeing that there are many deaths happening because of this COVID-19. And in our article, we are trying to highlight particularly those countries which are affected with the COVID-19 and what are the trauma they are facing and how we can manage this trauma, and how we can reduce the stress levels to keep our mental health stress free. So this is our point of our theme. So as I have told you that because of this COVID-19, the related updates and our news about coming through the media caused infodemic among the population, and this disease spread of this disease, there's a first like psycho social impact on the healthcare professionals, sudden rules and regulations due to the lockdown led to many economic depressions, physical distancing, quarantine among population. All this together affected the psychosocial impact, and the information dissemination. That is like people started, there are lots of false news, myths, and facts regarding the COVID-19. So this all came from the through the media, social media or Instagram, fake news that is being disseminated through the social media. This started creating some sort of panic among the public. And what the government agencies are trying to do is like they are trying to provide mental health education by daily updating about the COVID-19 and also bringing public awareness and regarding the mental health issues that is being associated with the pandemic. So inaccurate, you can say that you have to watch the official websites and internet and mobile device applications, which is giving you the precise information regarding the COVID-19. So there are several government who are trying to give psychosocial support, that is social support for you, that is being provided by the government as well as the private agencies. So what they are trying to do is they are trying to give you the psychological assistant to the team, like providing health services 24 hours service to the people. And there is also some psychological rescue team members or there who are trying to give advice to the people, educate them regarding the COVID-19, and the expert team or the people who are training the people regarding the health education and supervising them what to do, how to you have to educate this mental health persons. Basically, if you see the in the B diagram where I have shown you that who are the people who are facing a lot of trauma, the psychological trauma. The first level of the psychological trauma were faced by the frontline healthcare workers, and the people who participate in the frontline prevention and control of the epidemic disease. The second level is people who are self-quarantined patients who are regarded as a positive with the COVID-19, having a mild symptoms or no asymptomatic patients. So this suspected COVID-19 infected persons are facing a lot of stress. And third level is the people who are related to the above first and second means their relatives and friends, because their family persons who have been self quarantined, and the other people who are outsiders of the population, they started like creating much of the havoc, and because of this the stress levels have been increased. And the fourth level are the people who are affected by the prevention and control measures in the epidemic areas, or the susceptible population and the general public. So these are the different levels where the population having various mental health issues, there is a lot of initiative measures that needed to be taken by the national as well as the local government in coordination for prevention of this crisis. And the major thing that we have to focus here is that social distancing because of the COVID-19 is one among the reason that is causing the psychological trauma among the people. Whether you take it as old people or people with some sort of chronic diseases or children or teens. So the rise of the COVID-19, like the doctors who are attending the healthcare providers, so their mental health conditions started deteriorating because of the abuse of the population, other population members, because of the doctors who are attending those COVID-19 patients. They're prone to tell that they might the infection might spread to the other population also. So these are the strategies that is causing the psychological trauma. So what we can do is the World Health Organization has recommended the mental health people that you limited time you watch the news regarding the COVID-19 updates. Never constantly listen to the media coverage regarding the COVID-19 because that can cause you anxiousness and upset, and it can upset your mental health. And the this mental health trauma has started when the first wave of the COVID-19 has started, which caused immediate mortality and morbidity. And the second wave caused restrictions, that is a lockdown of the thing. And the third, all chronic conditions started proliferating. So these all caused mental illness and economic depression. So all these reasons have been accumulated or accumulated to form the main reasons for the psychological trauma. So what can we do to reduce such psychological or emotional states? That is the basic criteria that we have to plan. Each and every government agencies have to plan to manage that stress levels that is being caused because of the COVID-19. So we have to cope with the mental issues during the COVID-19 pandemic. So due to lack of regular social activities, we are because we are staying at home for a longer duration of the time, and our emotional well-being is also being changed. So the mental health conditions is being deteriorating. And one other reason that Ho et al. Who also have clearly portrayed that this sudden outbreak has caused mental health for new patients or new population, you can say. But those who are having the pre-existing mental health illness have also started, their condition also started to deteriorating. So the main impact to reduce the COVID-19 stress is that you should have adequate sleep, do exercise regularly, you use yoga techniques, relaxation techniques to cope up with the mental health challenge. So you have to change your lifestyles. So the coping during the COVID-19 pandemic is very, very important. So you go for relaxation exercise, yoga, meditation, aerobic exercise, prayers, or you can do some kind of creative activities. Positive thinking has to be there, or reading books, music. So whichever is applicable for you, you have to go for. And the young minds or young youth can go for online courses. So these are the things that you can cope. So what are the positive tips for the mental health during the COVID-19 pandemic is you should have your positive thinking, regular exercise, balanced diet. So diet is also important to boost your immunity, and socialization, that is online interaction, like you can attend lots of webinars or international or national webinars. So you yourself have to get engaged in lot of work and taking a break from your routine work life, other health lifestyle measures. So don't the don'ts or that they don't believe in the fake news. So you have to restrict yourself watching lot of news, that is the fake news especially. There are lots of myths are there, so you have to study the both the myths as well as the facts. And don't sit like couch potato. Don't be like a couch potato sitting in front of TV watching news and start eating too much of the fast food. So you have to exercise a lot. So don't focus on the negative aspects of the COVID-19 pandemic. Always stay positive. That is most important. The positive mental health will give you relaxation from any kind of mental illness. So you have to divide your work time. Take short breaks to relax yourself. A healthy routine lifestyle, boosting immunity, participate in several welfare works, conduct health education webinars. You can also act as a resource person in conduction of many kind of webinars, communication, association programs. In order to reduce the stress at a maximum rate. So the main reasons for the anxiety is that 40% of the people fear that your loved ones getting sick, fear of getting sick due to the pandemic, the worriness about losing the job, because due to the lockdown, many people have lost their jobs, the economic depression has occurred, the total GDP has come to down, not only in India but globally it has affected a lot. So the sudden lockdown in the March, during the time the India was the one who has started completely implemented with this lockdown program during the coronavirus pandemic. So what the management tips that I am trying to give you is that e-learning process. Go for Swayam or Swayam Prabha, or you can go for Coursera. So these are the online courses, NPTEL. You can have your career oriented, whether it be of any age people, do some sort of online courses to engage yourself, protect yourself from the coronavirus. So the main important thing that you have to do in order to reduce your psychological stress is you keep on washing your hands, and your elbow, cough or sneeze, you have to use your elbow, not your hands. Avoid touching your eyes, and social gathering should be limited. Don't gather yourself in crowded places. Clean and disinfect frequently. So these are the important tips that need to be handled. And the other important management strategy uh, that I am trying to highlight is that the AI technology, the development of the artificial intelligent technology, especially to combat the COVID-19. So we have to be grateful to the drones that is acting as a medical kits, and robots, 5G robots, which are especially utilized in the hospitals. Like they have the ability to disinfect the rooms and wards, and the multi-purpose robots, that is, they can provide food to the people who are in the isolated wards, who are affected with COVID-19. So the artificial intelligence made 5G robots introduction caused much relief for the healthcare workers, as well as reduce the stress level to a maximum. So as the novel coronavirus continue its ambush across the globe, so it instigates the use of this kind of multitude technological approaches like IoT, Internet of Things, so telecommunication networks, 5G to the frontline people. So even I belong to Mahila University, so we also took certain initiatives to help the people who are facing the psychological trauma. So we are providing COVID-19 help desk. So you any students can call us at this help desk and get the help. So our location we have shared, which is on lots of social connectivity like Facebook, Twitter, Instagram. So if all 24 hours the helpline is being open, and my good suggestion is that you avoid the body contact until the vaccine is being available. Now vaccine is being available. In India, we are using Covishield vaccine, which is now being Covaxin, Covishield, both are being available. Even Pfizer has introduced the latest vaccine. So lots of vaccines have been arrived, but still to reach the general population, it will take nearly September month. So till that time, avoid the body contact. So my basic conclusion is that
Dr.
COVID-19
Fatma,
carries significant.
your time
Yeah,
is
I'm
running
winding up.
out.
So this, yeah, just 1 minute. So despite these challenges we are all facing now, the COVID-19, so we have to strive to live by our values. So the important thing I am trying to tell you is that better immune system, good hygiene, spend at least 30 days outdoors, stay in touch while maintain social distancing. So these are the references that I have referred. Thank you.
Thank you for your presentation. You have questions?
Thank you.
Bilge Kağan hocam, sunum sırası sizde galiba. Tekrar bakayım. Sunumunuzu siz yapacaksınız galiba. 3 hocamız var. Mikrofonunuz kapalı hocam, Bilge hocam.
Tamam hadi. بعدك
Hocam Roa Hanım yapacaklar sunumu.
Tamam hocam.
Roa alsaydı.
Tamam hocam. Amra
Dr. Fatma, you are moderator. Hocamızın bilgisi yok galiba. Ra hocam siz buyurun.
Hello everyone. Can you see the presentation, please?
Yes.
Yes, you can start.
Tamam. Avian coronavirus infectious bronchitis virus IBV infection. A review from the COVID-19 perspective. by PhD student Roa Saidi and Assistant Prof. Dr. Bilge Kaatikal Olo and Prof. Dr. Yasemin Yanar in the Biotechnology Department, Çukurova University, Adana, Turkey. Infectious bronchitis virus was described in 1931 in the USA as the first coronavirus. It was then recognized as the cause of respiratory disease in chickens that could be transmitted via contact exposure, bronchial exudate, and via cloacae. A series of studies were developed, replication and modification of IPV virulence by subsequent passage in the embryonated eggs. The idea of immunizing flocks against IPV emerged from field observations showing that chickens infected with IPV prior to egg lay between 8 and 16 weeks showed mild respiratory signs and no adverse effects, becoming immune to the disease when they reached onset of lay. This was the starting point for the extensive immunization programs as currently practiced. The most widely used H vaccines were first developed in Germany by Dutch graduate students in the early 1960, being its most important exponent in ubiquitous H120. Initially, all the isolates were believed to belong to a single Massachusetts serotype, but in 1956, the authors reported isolation of second serotype, which is named Connecticut. Subsequently, other serotypes were described, and it is clear that a considerable number of different serotypes now exist. Despite several related pathogenic viruses, for humans, coronaviruses have been regarded to be mainly of veterinary interest throughout the 20th century. All non-human coronavirus during that time were associated with mild respiratory disease. In contrast, coronaviruses have been known to be the cause of more severe problems in veterinary contexts. IPV, for instance, has caused tremendous economic losses for the poultry industry for decades. With the appearance of severe acute respiratory syndrome, SARS, and a small-scale epidemic in 2002 to 2003 caused by the SARS coronavirus, the general interest in corona research was strongly increased. It was shown that SARS originated from bats and crossed the species barriers, indicating the potential threat of animal coronavirus for the human population. This notion was strengthened by Middle East respiratory syndrome in 2012 in Saudi Arabia by another zoonotic coronaviruses. The increase in coronavirus research after SARS outbreaks led to a large increase of knowledge regarding the coronavirus phylogeny. Given the high prevalence and wide distribution of coronaviruses, their large genetic diversity, as well as the frequent recombination of their genome, and increasing activity of at the human-animal interface. This virus represents an ongoing threat to human health. This fact again became evident in late 2019 when a novel coronavirus was discovered to be the cause of large and rapidly spreading outbreaks of respiratory disease, including potentially fatal pneumonia in Wuhan, China. The virus was characterized as beta coronavirus and recognized as the seventh discrete coronavirus species capable of causing a human disease. Avian coronavirus is a highly infectious and contagious pathogen of domestic fowl that replicates primarily in the respiratory tract, but also in some epithelial cells of the gut, kidney, and oviduct. Morphology, structure, and replication of coronavirus. The genome of coronaviruses are a group of large envelope, non-segmented, positive single-stranded RNA of about 26 to 32 kilobase, making it the longest RNA viral genome known. Taxonomy of avian coronavirus, the genus Gamma coronavirus, subfamily Coronavirinae, family Coronaviridae, order Nidovirus. The major serotypes of avian coronavirus include the Connecticut, Massachusetts, Delaware, Arkansas, Georgia 98, and QX type. The host of avian coronavirus is the chicken, naturally infected, respiratory. It causes two forms, include the respiratory form, more severe in chicks, and the critic form seen in birds under 10 weeks of age. The clinical signs in chickens. Within 24 hours post-infection, chicken infected with infectious bronchitis virus may begin to show signs of respiratory disease. The flock of layers is exhibiting a mixture of respiratory signs, including coughing, sneezing, and coughs. In the flocks of birds older than 6 weeks of age, these signs may be subtle and may only be observable at night when the birds are normally quiet. Baby chicks showing signs of gasping and respiratory distress. Other signs in young chicks often include coughing, sneezing, coughs, and oculonasal discharge. In most strains, mortality is usually negligible unless complicated by secondary bacterial infection. However, chicks less than 2 weeks old that have a natural IB infection or a severe reaction to IBV vaccine may suffer permanent damage to the oviduct, resulting in a poor to no egg production capacity later in life. The other clinical signs include mucopurulent ocular discharge associated with the conjunctivitis, ocular involvement in most commonly observed in young chicks, and may include epiphora, tearing, and conjunctivitis. When the egg is opened, the quality of egg is often found to be inferior. As seen in the egg of the left, the albumin may be thin and watery, and the separation between thin and thick albumin may be absent. A normal egg is shown on the right for comparison. When infectious bronchitis virus replicates in the reproductive tract of layers, normal calcium deposition in the egg shells may be disrupted, resulting in soft, misshapen eggs. In layers, egg production may drop by up to 50%, and eggs may be soft-shelled, fragile, and misshapen, as seen here. The pathogenicity of infectious bronchitis virus can vary widely between strains. The clinical outcome of an infection in chicken depends on many variables such as the virus strain and type, sex and age of the chicken, immune status like vaccination, immune suppression, and maternally derived antibodies, co-infection, and environmental circumstances such as climate, dust, ammonia, and cold stress. Infectious bronchitis virus, gross pathology. Infected chickens have cirrhosis, catarrhal, or gaseous exudate in the trachea, nasal passage, and sinus. Air sac may be foamy during the acute infection, and then may become cloudy and contain yellow gaseous exudate. Areas of pneumonia may be observed around the large bronchi, The nephropathogenic infection produce swollen and bell kidney when with the tubules and ureters often distended with urated. Fluid yolk material may be found in the abdominal cavity of chickens that are in production, but this is also seen with other diseases that cause a marked drop in egg production. Cystic oviduct may be a consequence of IBV infection at a very young egg and are a cause of reduced peak in egg production when reaching maturity. Effect of IBV infection on the reproductive tract of chicken in production have been detailed by Sivion and Levin, who observed the reduced length and weight of the oviduct in infected birds as well as regression of the ovaries. Diagnosis of the infectious bronchitis in chickens include on the basis of symptoms and lesion, isolation and identification of virus, samples to be collected from trachea, bronchi, lung, kidney, and cecal tonsils. 9 to 11 days old embryonic chicken egg inoculation via endotoxic routes. After 3 to 4 days, curling and dwarfing of embryo is seen. Antibody detection by virus neutralization test, hemagglutination inhibition, immunofluorescence, and ELISA. SARS-CoV-2. SARS-CoV-2 is a part of the coronavirus family, as is SARS-CoV-1, which generated the 2003 SARS epidemic, and MERS-CoV, which generated the MERS epidemic in 2013. The new SARS-CoV-2 coronavirus, severe acute respiratory syndrome coronavirus 2, causes a respiratory infection called COVID-19 coronavirus infectious disease 2019, with an average incubation period of 5 days, between 2 and 14 days. The disease is characterized by predominant respiratory symptoms, include cough, difficulty breathing, and fever of moderate intensity in about 80% of cases, but may have severe manifestation, include bilateral interstitial pneumonia with progression to respiratory failure, acute respiratory distress, the main cause of mortality. There are also cases with gastrointestinal manifestations, include especially diarrhea, and in some patients, especially in young ones, hypoosmia, loss of sense of smell, and hypodysgeusia, alteration of sense of taste, have been reported as early symptoms. SARS-CoV-2 virus structure is composed of an RNA molecule surrounded by a series of structural and functional proteins. Non-structural proteins are protein S or spike protein, which lead to a characteristic appearance, membrane protein, envelope protein, and nucleocast protein. Of these proteins, S has the role of attaching to receptors in human cells and facilitating the fusion of viral content with the cell. In the case of SARS-CoV-2, the receptor is the angiotensin converting enzyme 2, which is found in the large quantities in the respiratory tract and lung parenchyma. The initial infection of SARS-CoV-2 occurs in the ciliary epithelial cells of the bronchi. Potential therapeutic strategies against COVID-19. Initially, interferon alpha nebulization, broad-spectrum antibiotic, and antiviral drugs were used to reduce the viral load. However, only remdesivir have shown promising impact against the virus. Remdesivir only, and in combination with the chloroquine or interferon beta, significantly blocked the SARS-CoV-2 replication, and patients were declared as clinically recovered. Various other antivirals are currently being evaluated against infection, include nafamostat, nitazoxanide, ribavirin, benciclovir, favipiravir, ritonavir, and arbidol exhibited moderate results when tested against infection in patients and in vitro clinical isolates. Several other combinations, such as combining the antiviral or antibiotics with traditional Chinese medicines, were also evaluated against SARS-CoV-2 induced infection in human and mice. Recently in Shanghai, doctors isolated the blood plasma from clinically recovered patients of COVID-19 and injected it in the infected patients who showed positive results with rapid recovery. History of vaccines. Pardon. History of vaccines for coronavirus. Coronaviruses have a large single-stranded positive-sense RNA genome encased by helical nucleotides and an outer envelope comprised of matrix protein M, envelope protein E, and spike protein. The spike protein, which naturally occurs in trimeric form, it contains the receptor binding domain responsible for binding onto the angiotensin converting enzyme 2 and entry into the cell. In SARS-CoV, of all the structural proteins, spike protein was found to elicit neutralizing antibody and is major target antigen for vaccine development. There have been difficulties in the development of coronavirus vaccines historically. Coronavirus vaccines in animal models that mimic human disease have been immunogenic, but generally not shown to effectively prevent acquisition of disease. Further, there is a concern that vaccination, as with natural coronaviral infection, may not induce a long-lived immunity and reinfection may be possible. Mice vaccinated with two inactivated full virus vaccines, a recombinant DNA spike protein vaccine, or a virus-like particles vaccine, develop lung pathology, including eosinophil.
Hocam toparlayabilir misiniz?
Pardon?
Süreniz bitti.
Tamam.
Hızlı anlatabilir misiniz?
Tamam. The second slide. Vaccine candidates as of the 1st June 2002, there are currently 124 candidate vaccines that are under development for prevention of COVID-19 listed by the World Health Organization landscape summary, of which 10 candidate vaccines specifically designed for prevention of COVID-19. Have entered phase 1, combined phase 2, phase 2 human clinical trials in adults. Most of these trials are enrolling healthy adults from age 18 years only, or with the upper age limited of inclusion ranging from 50 to 60 years. Two trials are enrolling young participants, one from age 3 years or other 6 years, with no upper age limits in both. One combined phase 1 or 2 trial include older adults up to age 85 years, while another early phase 1 trial has been extended in May 2002 to include also older adults. This is the reference we are used. Thank you for your attention.
Çok teşekkürler hocam sunumunuz için.
Sorry. Hello.
Hello.
Oui. I have just a remark to Roa. If I think if we use or you use some table and some graphs in your research, it is a good thing because
But I didn't do يعني the research, but it is a review.
For a review, we can use table and graphs. For example, for slide 19. No. 19. You can give a table with a graph. We understand more than we use the phrase. Okay.
I used it تمام.
Sorry.
Inshallah in the future I will do it.
Okay. It is just a remark. It's because I am a statistician and we have a lot of data in coronavirus, so we can use those data in our research. Okay, it's a good thing. Okay, thank you.
Thank you very much.
Teşekkürler. Özgür hocam, buyurun hocam.
Okay. Hello, everyone.
Hello.
Let me first open my share file. Okay, so first I would like to thank to the organizers of this Congress. And I am really happy to have these presentations. Actually, I'm going to summarize our studies related to non-small cell lung cancer, which we are studying in our laboratory in Adana Alparslan Türkiye Science and Technology University, Adana, Turkey. And my first presentation will be related to the integrated bioinformatics analysis for identification of novel biomarkers and drug targets in non-small cell lung cancer. Actually, this is the outline of my talk. I'm going to present you some theoretical background about lung cancer and non-small cell lung cancer, and what are the current biomarkers in NSCLC, which is non-small cell lung cancer, obviously. And then I'm going to present the aim of the study and methods, our methods for the bioinformatics analysis. Then our materials, which are the microarray datasets, and then some results that after the bioinformatics analysis of these microarray datasets. Lung cancer is actually the formation of malign tumor mass in lungs. It is very, very oversimplified definition for the lung cancer, actually. And this is caused by the uncontrolled proliferation of cells in the normal lung tissue. So this is like normal cells go crazy, let's say. And when we look at the World Health Organization data about the estimated number of cancer cases, the lung cancer, the incidence of lung cancer is the first place in terms in men, and it is the third place right after breast and colon cancers in women. And when we look at the mortality rates of the lung cancer worldwide, when we combine the both sexes and all ages, the lung cancer ranks the first in cancer-related deaths, and it is the first in men and comes after the breast cancer in women. So it is a really dangerous, deadly disease, actually. And when we look at the histological classification of lung cancer, we see basically three types: the small cell lung cancer, non-small cell lung cancer, and lung carcinoid tumors. And actually the non-small cell lung cancer, which constitutes the 85% of the all lung cancers, includes the adenocarcinomas, squamous cell carcinomas, and large cell carcinomas histologically. And when we look at the treatment methods for the non-small cell lung cancer, according to the tumor node and the metastasis or TNM classification, we can classify the lung cancer tumors or staging. It's called, when we do the staging, we have several stages of the disease. And actually, because lung cancer, I'm not going to go into detail of that, but because it's a special disease, the surgery, which is possible for other cancer types, is not always possible in this type of cancer. So it leaves us with only chemotherapy options, and then basically the chemotherapy is used. And that means that we need a lot of drugs and new drugs for the treatment of non-small cell lung cancer. And nowadays, if you see the stage 4A, 4B, which is the advanced stage of the NSCLC, it is possible to have the targeted therapy, which is also called the personalized medicine. And the therapy for person, we can say, and the immunotherapy, nowadays it's very convenient for the treatment of the non-small cell lung cancer. And when we look at the biomarkers in the NSCLC research, actually these biomarkers can be used to first to diagnose the disease, decide about the histological types, and as well as decide about the treatment options. But if you look at the chart, you see 31% of, or 1/3 is unknown. So there are no certain biomarkers that you can diagnose the disease or follow up the treatment in the patients. So that means that we need new biomarkers for treating the NSCLC patients. So that's why there's a high demand for identification of novel biomarkers. And we need an enhanced understanding of the molecular biology of this disease for better diagnosis and treat the non-small cell lung cancer patients. That's why the aim of this study is to identify some novel prognostic biomarkers and their potential treatment drugs, not only the biomarkers, but some suggestions for the potential treatment for these biomarkers by utilizing the public omics databases and then so-called integrated bioinformatics approach. This is the flow chart of the workflow of our study. So first we collected the data from microarray datasets, the NCBI gene expression omnibus database. And then we pre-processed the data with some statistical methods and some web tools like R bioconductor and then robust multi-array method, as well as the Limma method. And then we identified the differentially expressed genes and then found their biological meanings by utilizing the functional and pathway enrichment analysis. Also, we used some consensus path database. I'm very sorry. Okay, I will continue. I'm very sorry. So we use some other databases to find the biological meaning of our data. These differentially expressed genes, what are they doing in the cells? Then we analyzed some protein-protein interactions, which is like proteins that can affect the other proteins and cause some major pathways changes in for the development of the disease. And we check the prognostic performance. So what happens to the patients when there is a differential gene expression in a patient? And then if these protein-protein interactions, so-called hub genes, change, what happens to the patients? We analyzed with some survival analysis. And then finally, we analyzed the drug target potential, which means we offered some novel drugs to change the expression of these differentially expressed genes to return them back to the normal expression. So these are the genome, the data, microarray data sets we found, and about 200 tumor samples versus some control samples we get from the GEO database. So tumor versus adjacent normal samples. And then we analyze the raw data for like pre-processing the data. And then using the R bioconductor platform and some robust multi-array and limma methods we combined, and comparing the gene expressions in NSCLC tissues with some neighboring healthy tissues, we identified about 1,000 common differentially expressed genes in these 4 data sets. And then 246 of them were upregulated and 734 were downregulated, as you can see in this Venn tables. And then to find out the biological meaning or biological pathways that these genes are related to, we looked at the biological insights of the common transcriptomic signatures. And then we found that these DAGs belong to some pathways like cell cycle and P53 signaling pathway, which is important in the cancer development, and some extracellular matrix receptor interactions, which is important in the cellular immunity, actually. And these were related to the upregulated genes in this gene sets, DAGs, so-called the differentially expressed genes. On the other hand, on B, you see the downregulated common DAGs and 32 terms, including some unusual malaria, cell adhesion molecule, CAMP signaling pathway, etc., related, or the genes that downregulated are related to these biological processes. And when we construct the protein-protein interaction network, so we are like hunting the critical proteins that may play a role in the development of the NSCLC. So that's why we needed to analyze the important proteins, or to figure out the important proteins. When their expression changes, then they change the expression of other genes. And those genes are called the hub genes. And using this protein-protein interaction network, you can reach to hub genes, actually. So we analyzed the proteins from our data, and we found that some candidate hub genes, like these degree score and betweenness score, are like used to highly connected proteins. That means like they are related to more protein pathways, signaling pathways. So like CBL, EZH2, CDK1, CDC20, etc., found, and these are the top 10 hub genes we figured out. And we checked the prognostic performances of these identified hub genes. And on top line, you see CDC20, ORCA, CDK1, EZH2, CDKN2A. These were when the mRNA expression of these genes increase, so it is bad for the overall survival for the NSCLC patients. But below the EGR, CBL, etc., below line shows that some among the top 10 genes, when their expression is high, which is good for the survival of the patients, so we can say. And this is the proof of the concept kind of data, because those genes that are bad when they are upregulated seems to be upregulated in different data sets here listed, and they are shown as red. And the blue ones, actually, our cutoff value is like 0.5, so below 0.5 means that downregulation. So they seem to be downregulated in all data, and they are shown in blue. And finally, we use the LINCS 1000 connectivity map data with some signature search engine, which is called the L1000 CDS. We assess the potential drugs that may reverse these expressions, whether if they are downregulated or upregulated. What should we use to return them back to the normal expression? So we identified some potential drugs by using the signature search engine, and a considerable portion of these drugs are anti-neoplastic, which are useful for the cancer treatment, as well as we see some other drugs which were originally used for other purposes, but somehow we were able to find out some alternative drugs to potentially change the expression of these targets. And this is all about my first presentation.
Özgür Hocam, çok teşekkürler sunumunuz için.
Kusura bakmayın, özür diliyorum. I'm very sorry that because I had some sort of trouble here, so I was not able to talk. So I'm ready for your questions.
Hello. Yeah.
Yeah, I can hear you.
I have a question, please.
Of course.
Thank you. For some graphs, you have used minus log p-value.
Yes.
Why? I don't understand why minus p-value, minus log p-value for some graphs.
Okay, I will check that.
Why we don't use p-value without minus log?
Yeah, okay, I understand. There's some log rank and HR values. I think you are talking about this survival analysis. Actually, I should say that we are using some web tools in that case. In this analysis, we apply the data to our candidates to the web tools.
Please, can I see your?
Of course. I'm sorry.
Thank you.
I'm sorry. Okay, I will show you. Hold on a second. Screen. This one. Okay. Can you see?
No. If it's not possible, no problem.
Okay. Okay, I think now it's okay. I wonder which data are you talking about?
Even,
Minus log.
yes, here, no, here, for example, here.
This one.
Mhm.
Yeah, actually, I was trying to explain this one. This comes with like kind of the result of the, how should I say, the web tool calculates these values, the relatedness of the sample to the population, let's say, and they have several populations. And these are obviously the biological pathways. I'm actually don't know why they give this minus log p-value. I'm just interested in the table, the names of the numbers below means that they are more related or more genes related to the process.
Okay.
So
Thank you.
the blue line, which is important for me.
Because me also, I don't know why we have minus log p-value.
Yeah.
It's, okay, thank you.
Shows the number of processes.
Okay, thank
so
you.
that's why we use that.
Thank you.
Okay, you're welcome. Okay, then I can continue with my next study. It's more, actually this was, the previous study was more like numerical study. So I am really new to that, but my specialty is in the molecular biology laboratory. So this is more related to my work actually. We try to model the drug resistance in non-small cell lung cancer by some in vitro studies with my previous graduate student, Aylin Gündüz. And okay, I can jump forward actually because there are same things up to drug resistance mechanisms in NSCLC, why they are important. I talked about the lung cancer, why it is important, the high death rate, and then the non-small cell lung cancer and staging. Although the chemotherapy seems to be like the major option to treat the non-small cell lung cancer, intrinsic or acquired resistance, which means that intrinsic means that previously present in the lung cancers. And acquired resistance is something like that on the way, the cancer cells get the resistance against the drugs, actually. They developed some resistance against chemotherapeutic drugs. And because of that, the survival rates in these NSCLC patients remains low. So we have a chemo resistance or drug resistance problem in NSCLC treatment. So actually, when we look at the chemotherapy drugs for NSCLC treatment, we see platinum-based chemotherapy, which is very common actually in other cancer treatments because it is systemic and it's a general treatment strategy, as well as some targeted drugs, as I mentioned previously, I guess, targeted drugs, personalized treatment, like certain mutation and certain drug that can cure that mutation you can think of. So erlotinib, gefitinib, and afatinib is that kind of targeted drugs because the EGFR, the epithelial epidermal growth factor receptor mutations are common, very common in NSCLC. So this causes some really big problems, increased expression of EGFR there, and then these inhibitors, they block the expression of EGFR so that they try to cure the NSCLC. But the drug resistance, as I mentioned, there are two types, intrinsic and acquired resistance, is the ability of cancer cells to survive and grow in the presence of multiple and variable drug treatments. And it's a major pharmacological problem in the clinic because you give the drug to the patient, but the patient somehow does not respond to that drug. The intrinsic resistance mechanisms, as I mentioned, is basically related to the EGFR mutations, which is very important pathway actually in the growth of the epidermal cells. But somehow mutations on this signaling pathway causes lots of troubles in the NSCLC cases. And it is instantaneous ineffectiveness of the drugs against this EGFR. And there are some point mutations, insertions, deletions, and several changes in the EGFR gene. And then there are drugs for these intrinsically resistant NSCLCs. And although some of the patients have EGFR activating mutations, they cannot initially provide the desired clinical outcomes, which means that even if you give the drug, it doesn't work there because of the listed reasons. Acquired resistance, however, it is something gained during the treatment. But if you treat the culture cells with drugs in vitro for a long time, secondary or acquired resistance is formed. That means that even if you give the drug, in the beginning there were some responses, but later on you can get that response. And the resistant phenotype can appear through a number of molecular mechanisms. And as you can see, these are somatic mutations. And then there are different types of somatic mutations which may cause the acquired resistance mechanisms. And actually, for modeling the drug resistance, especially the acquired resistant mechanisms, there are two general models. Why we need these models? Because we need to understand the drug resistance mechanisms more in detail, and we can develop some new approaches and some new drug treatment strategies. So we have, for modeling the drug resistance, we have clinically relevant models and high laboratory level models. Actually, a basic difference is that clinically relevant models use low amount of drug and more time, just like the chemotherapy cycles in the hospital. High level laboratory models use like high dose drug administration, back to back. In time, you increase the drug concentration, and you can observe more molecular changes in this case. So we can use cell lines, animal models, organoids, spheroids, etc., in modeling drug resistance. But in our laboratory conditions, the easiest one is the two-dimensional in vitro models using the cell lines. Erlotinib, as I mentioned previously, one of the chemotherapeutics used to treat the NSCLC patients, and it is a tyrosine kinase inhibitor for the EGFR, which itself is a tyrosine kinase. And its commercial name is Tarceva, and it is molecularly targeted drug used in NSCLC patients with EGFR mutations. And unfortunately, NSCLC patients develop resistance to erlotinib in 10 to 14 months after the primary therapy with the drug. So the aim of this study was to establish and characterize the EGFR TKI erlotinib resistant NSCLC subcell lines. So we provide drugs back to back, increasing the concentration of the drug in vitro in culture, and then check for the results. And here are the methods. We choose a cell line A549, which is a non-small cell carcinoma cell line, frequently used in drug development. And this is our cell culture methodology, just towing the cells, subculturing, and seeding in appropriate numbers. Apply the drug, wait for the cells to grow, again treat the drugs. And we check the appropriate cell number for seeding the cells in different cell culture plates. We tested them. And then in order to create or establish some erlotinib resistant cell lines, we first checked the theoretical data, which is the erlotinib concentration. which will be important in developing some resistance. And then we search for the appropriate starting dose of erlotinib, and then choose the parental cell line, increase the erlotinib doses in time, and then some with treatment intervals, which is 2 weeks actually. And on the other hand, we treat some parental cell lines in equal volume of solvent, not the drug, but I think the doctors, they call this placebo. And drug concentrations expose the cells adjusted by mimicking the treatment conditions of cancer patients. And our project was based on peak plasma concentrations and IC50 values of the drugs provided from the publications. And concentrations are determined such that these values remain between the concentration values for maintaining some pharmacokinetic properties, which will be important. But because if you I mean that you increase much more than the physiological condition, it's meaningless. So we should keep it in a pharmacologically feasible manner. And then from the literature, we figure out the IC50, the calculated IC50 for our cell line is about 6 micromolars. And then we decided to increase the concentration by 3 micromolars each time, and then per 2 weeks intervals. And this is what we did is we treat the parental cells with some solvent, which we put the erlotinib drug inside, and then per 2 weeks we increase the erlotinib concentration, subculture our cells, and continuously, like about in 2 months, up to 12 micromolar, which is 2 times more than the feasible value. And then at the end, for another month, we kept our so-called ER cells, which are erlotinib, which stands for erlotinib resistance. We kept them another month. Finally, we got some candidate drug resistant or erlotinib resistant cell line. After that, we checked the cell viability of our parental and resistant cell lines using the MTT method. And then we checked whether if our cells are migrating or invading the neighboring areas by wound healing assay, because aggressive tumors, they migrate faster, stronger. So we needed to check that whether if our drug treatment just increase the aggressiveness of our cancer cells. And then we check the colony formation because high colony formation means, again, more aggressive tumors, whether if the drug treatment is increasing the colony formation ability of our cells. And finally, we perform some quantitative reverse transcriptase PCR, just isolating the RNA and converting this mRNA into cDNA with some reverse transcription, and check the qPCR by using cybergreen dye, and then analyze our data by using delta CT method. And these are the results. First, the drug treatment changes the morphology of the cells. This will be important in the understanding the later on results, because we check since the morphology is changing into somewhat a mesenchymal type, we check whether the molecularly cells are changing their gene expression towards the mesenchymal cell type biomarkers. And this is the result of our cell viability assay. Actually, we do not expect much in this one because our cell line is a EGFR wild type, and then the drug was designed to mutated EGFR cell lines or cells. So we do not expect much in this. However, when we check the cell wound healing assay, the drug treated cells or the resistant cells, although there is high range of the mistake or errors, the standard deviation is too high, I can say. Still, they migrate, the erlotinib resistant cells migrate faster than the parental cell line in different intervals. And in this figure, we see the colony formation, the tumor formation capacity. So the treating the cells with the prolonged exposure with the erlotinib increases the tumor formation capacity in the cells. You see on top A, here in this figure, the ER at the end of ER and A549 ER. So this is erlotinib treated resistant cell line, forms more colonies, which means that tumor formation capacity in these cells increases. And it is significantly, statistically significant. And finally, our qRT-PCR results that unfortunately only in one CDH2, which is N-cadherin, and which is a sign of mesenchymal cell type. As I mentioned previously, the morphology change into mesenchymal cell type, only one indication or one gene expression in accordance with this morphology change we could find. And we still continue analyzing the cells with other markers. So our preliminary conclusions on this study, besides the enhanced cell viability under drug conditions, erlotinib resistant subline was more tumorigenic with a greater migration capacity than the parental subline. And the mesenchymal cell type biomarker N-cadherin, the CDH2, as you had seen in qRT-PCR data, was significantly increased in erlotinib resistant subline. And in future, we hope to do some microarray or RNA sequencing analysis on these resistant cells to figure out whether if there are other candidates of this, the reason for this increased migration capacity or tumorigenic capacity. Okay, thank you very much for listening my presentation. And if you have questions, I will be happy to answer your questions.
Özgür Hocam çok teşekkürler sunumunuz için. Özgür Hocam sizden moderatörlüğün devamı için rica edebilir miyim? Doktor Fatma Hocamız sanırsam yapmayacak. 4 sunumumuz kaldı hocam. Ben programda
Tamam.
da oturum başkanı olarak değiştireceğim şimdi.
Tamam.
Buradan sonraki 2 sunum hocam Zekeriya Mennane hocamız yapacak.
Okay.
12'şer dakika verebilirseniz hocam. Sunum süremiz de az kaldı.
Tamam.
Yani 2 sunum için ortalama bir 22 dakika yeterli olur hocamız için.
Okay.
Teşekkürler.
Okay, so I will be moderating this session. So please, Dr. Mennane, I invite Dr. Mennane for his presentations. Dr. Mennane, please.
Okay, thank you. For antimicrobial activity of essential oil and some extra of olive and some from Nimlal in Morocco. Okay. Olive tree is a specific tree of the Mediterranean basin whose likely origin is Egypt, India, Syria, or Ethiopia. Its cultivation in North Africa existed before the arrival of the Romans. The production of its wild in North Africa was recognized about 7,000 years ago. In Morocco, the production is about 2 million tons in years, and for area about 1 million hectares. The most widely available variety in Moroccan is Zitoun Bildi or Moroccan Picholine. The regions in Morocco which highest production of olive are Marrakesh, Safi, Bni Mellal, Fnifra, Tangier, Tétouan, and Fès Meknès. Medicinal plants have been used for thousands of years in view of their active component have efficacy in the treatment of disease, constitute a potential reservoir of effective antibacterial molecule. The first formal report of medicinal use of olive extra was in 1874 for treating malaria and fever. Photochemical investigation announced that olorpaine was extracted from the leaves of Olive europaea. This compound is known for possessing broad range of pharmacologic and health promoting properties, including antiarrhythmic, spasmolytic, immunostimulant, cardioprotective, hypotensive, anti-hyperglycemic, antimicrobial, and other. The report describing antibacterial properties of phenolic compound is olive productor refer to compound obtained from olive fruit, especially hydroxytyrosol and oloropaene. The aims of this work was to investigate the antibacterial activities of essential oil and some extra ethanolic extra and olive mild waste water of olive collected from Bni Mellal in Moroccan against gram-positive and gram-negative bacteria and also some yeast. Olive leaves and olive mild waste water were collected from the Bni Mellal region in Morocco. The leaves were cleaned from extraneous matter and properly washed, then dried at room temperature for 50 days. The dried leaf material were ground to fine powder using an electric grinder and stored in airtight in dry place until extraction. For extraction of oils used Soxley, this method by using the solvent of acetone. After you use rotary evaporator. Also for organic extra, use the same material.
Excuse me.
Yes.
Yüksel hocam, mikrofonunuzu kapatabilir misiniz acaba?
Pardon, hemen kalkıyorum. Çok özür dilerim.
I'm very sorry.
Yes. For bacteria, you use 9 bacteria: Streptococcus pneumoniae, Staphylococcus epidermidis, Staphylococcus aureus, Acinetobacter baumannii, Pseudomonas aeruginosa, Escherichia coli, Klebsiella oxytoca, Serratia marcescens, Proteus mirabilis. Donc, for yeast, you use Candida tropicalis and Candida albicans. These strains were collected from the National Institute of Health, Rabat, Morocco. The antibacterial activity were performed using the diffusion method on agar media, Moyer-Hampton, and saboro-dextrose agar for yeast. Organisms have been spread over this growing media by cotton swab. After well of 6 millimeters diameter were punched into the agar medium and filled with 15 microliters of plant extra. The plates were incubated for 24 hours at 37 Celsius. Antibacterial activity was evaluated by measuring the inhibitory zone against the test organism. Also, for minimum inhibitory concentration, you use this method MTT performed according to the micro-titration technique described by Elof. For result, for Enterobacteria gram-negative, the most activity were measured by in essential wear, while second is the olive oil wastewater, and the third is ethanolic extra. The most activity 38 for Klebsiella oxylotica. For no Enterobacteria gram-negative, also while essential oil is the first with the good diameter. The second is water waste of olive, and now result for ethanolic extra. For gram-negative positive, also the most was the essential wear, had no activity for ethanolic extra. For Candida, the essential wear while gave the good activity with 34 for Candida albicans. It's some photos for Escherichia coli, Serratia marescens activity, Staphylococcus aureus, also Staphylococcus epidermidis. For water of oil, the essential oil had strong antibacterial activity against Streptococcus 32 mm with minimum inhibitory concentration 155 microgram per milliliters. Also, same result for Klebsiella oxylotica. The olive mile wastewater was active against gram-positive bacteria, more than gram-negative, no active against yeast strain, and active for Staph aureus, Staph epidermidis, Acinetobacter baumannii. The extra obtained with ethanolic inhibited some bacteria and the zone of inhibition of growth. 40 millimeters against Klebsiella oxylotica and Proteus mirabilis. Other research indicates that olive composed of phenolic compound, especially hydroxytriazole and oleuropein, can be used for the treatment of various infections and for the development of new antimicrobial agents. This study, in conclusion, for this communication, this study emphasizes the antibacterial activity of essential oil and some extra, ethanolic extra, and the olive mile wastewater for olive collected from Beni Mellal in Morocco against human pathogenic bacteria. The maximum inhibitory zone was noted against Streptococcus pneumoniae and Klebsiella oxylotica. The most activity observed from essential oil, the second olive mile, and in the third ethanolic extra. Olea europea, composed of phenolic compound, especially hydroxytriazole and oleuropein, can be used for the treatment of various infections and for the development of new antimicrobial agent. Donc, for the first communication.
Okay, thank you, Dr. Menane. Actually, I personally have a question for you. Did you get any chance to have some GC-MS or mass spectrometry results for, checking the ingredients, the chemical ingredients of these ethanolic extracts or the other extracts?
Not yet. The first in this study, in this year, we used some material such as adsorption atomique, ou bien donc for
okay.
diagnostic of.
I see. And I think gram negative to gram positive, the effect is different. You said, I think it is quite important. As far as I know, some of the pathogenic bacteria is gram negative actually. So if find some real good chemical there, it will be really a very nice scientific meaning for that, I think, because we need antibiotics actually in future. They are becoming resistant. Okay, thank you. We can continue with your next presentation if no questions.
For ethnobotanical, the second communication for ethnobotanical study of fig tree and olive from Tétouan province and study of their activity antimicrobial. Fig tree is considered as a medicinal plant that is very old in Morocco. However, in some area, Taounat is the first province in Morocco for production. The second Chaouen and Ouazzane. Also, there is dried fig. In Moroccan nomination, Shriha was extracted with cereal. Its domestic production in 1940 was estimated as 156 millions of tons. The Moroccan is the third in the world. Turkey is the first, has the first production. Studies have reported the use of traditional medicine fig trees in different regions in Morocco. Olive oil is rich with polyphenol that have antioxidant properties to reduce free radical, especially the action of oleuropein. The content of phenolic compound is olive leaves reaches up to 250 milligram 4 gram matière sèche. These products are characterized by their antibacterial activity. The objective of this work is to study the traditional use of fig and olive tree in the region of Tanger, Tétouan by question, and study the activity, the antimicrobial activity of these plants. Study of fig and olive tree was carried out in the Tanger, Tétouan, Al-Hoceima region. The different points of investigation according to the administrative redistricting of the municipalities existing in the cities that were studied. Each question sheet has two components. The first related to age, sex, education, the origin of the information, the season, and the source. The second related to the part used in the plant, the type of disease treated by each part of the plant, and the method of preparation of some produce. Different distribution of study seat. We studied with Bni Hassan and Khamis and Jara province, Bni Ahmed and Talambut province of Chefchaouen, Malasso Delaya province and Tanger. It's the card of this region and the different cities. After all data in this study were recorded on the questionnaire sheet and then transcribed into the database, then processed by Excel software. For the antimicrobial assay, was performed using the diffusion method on agar media, Moyer Hampton and Saburo for yeast. Also, organisms have been spread over these growing media by cotton swab, and the well were punched into the agar for about 50 micron of plant extra. After 24 hours of incubation at 37 Celsius, the diameter of the inhibition zone was measured. The manipulation was repeated 3 times. For bacteria, used is Staph aureus, Pseudomonas, Klebsiella pneumoniae, Escherichia coli, and Candida for yeast, albicans. Ethnobotany is considered a study to acknowledge local population and their relationship with plants. The results obtained from this ethnobotanical survey on the traditional using of fig and olive tree in medicine in the region of Tanger, Tétouan, Al-Husima. The results for indicate that for the male, the most person was or male, and for age, the most is or high of 40. For level of population, the most have primary level. For agriculture or culture of the olive tree, the most has or the agriculture derived from olive consumed. The most population use olive oil for consumption, and for extraction method, the modern system is the first for this population. For medical use for the most use it olive and olive for respiratory disease and digestive disease, and in the third, the nervous system. For using of the leaves, the most use it in this region. For mastery of preparation, also the most population do it. For using the olive residue, the most population use it. For fig tree, the same of olive, the most are male, and the age is high for 40. Also, the level scale is primary. For situation married, the most is married. For origin of information is from experience, treatment and experience. People who have primary education were the most investigate in this study, with 56 and 77 respectively. Only 30 of farmer grow olive tree because it takes years to start production. 88 of population prepared olive in this home in different way. In this region, 62 use machine for extraction. Therefore, the traditional system does not exist. 38 due to the benefit of technology systems such as the performance and processing capacity. 52 do not use olive snack. 28 sell it to traditional oven, and 12 use it for livestock feed. The origin of information, the majority of fig tree depended is its achievement in traditional medicine. Most farmers use olive oil, and 50 of which 30 use it for respiratory disease, lubrication, the chest and neck in case of cows. Those who use in for digestive system, people who use it as a treatment of nervous system reaches 30. 33 use as treatment of certain abnormalities of organ function. The survey showed that 42 of farmers use olive leaf to treat certain diseases such as diabetes when cooking olive leaf in water and drinking the product after filtering. In addition, 30 use it for livestock feed. The result of activity antimicrobial and in this table for ethanolic extra and aqua extra of leaf of olive in different mountains. You look for stuff, the most is in October, and also for aquas extra stuff or use. For E. coli also the most diameter is in October. For Pseudomonas December, and for Pseudomonas Klebsiella candida the most is in December. For leaf of Ficus carica, ethanolic extra is such as ethanolic extra of leaf of olive. For aqua extra, there is 0 for different bacteria and yeast. The ethanolic extra of leaf of Ficus carica was active against all bacteria tests and yeast. Also, the ethanolic and aqua extra of olive leaves from Tetouan region was active against all bacteria tests and yeast by maximum inhibitory zone against Staph aureus and 53 and 12. All bacteria tests and yeast by maximum inhibitory zone, Staph aureus, E. coli, and Candida albicans 54 and 12. In conclusion, the ethnobotanical study of fig and olive tree in the various states of Tanger, Tetouan, Hoceima region of Morocco has collected information on the different use in traditional medicine of those two plants. The fig tree has been used against asthma, colds, coughs, constipation, and warts. So for the olive tree, most farmers use olive oil to treat respiratory disease, digestive system, nervous system, and get rid of constipation. air infection, and other. The results of antimicrobial activity indicate that ethanolic extra showed good inhibitory effect against most strains of bacteria. And thank you.
Okay, thank you very much, Dr. Mennane. Is there any question?
Yes, please.
Yes, Dr. Mehdi, please.
Yeah, I have just a remark for Mr. Minnan. If you use for your data, if you use the technique experimental design, en français ce sont les plans d'expérience, you will have a good results and interested results for your study. So I recommend you to check for experimental design, and you will find a good results for interaction between oil and feed, and relation between disease and oil and feed. So you will have a good study in this case. It's recommended.
Yes.
Thank you.
Yes.
Thank you.
Yes.
En français, ce sont les plans d'expérience pour chercher.
Oui.
Les plans d'expérience. Les plans d'expérience. When we have a repeated experience, a repeated user in an experience, we use the experimental design. Les plans d'expérience, quand on a les mesures répétées dans des expériences. Which is your case.
Pour la relation, oui, pour la relation entre les extraits, donc
yes.
la relation entre les composés de figuier et les composés d'olivier, une étude réalisée donc donc un laboratoire en Égypte et un laboratoire au Japon. Donc ils ont travaillé sur certains extraits. Ils ont trouvé vraiment il y a une relation entre, surtout pour les personnes âgées. Lorsqu'on consomme par exemple une quantité de figues, par exemple une seule, donc c'était dans le Coran avec cette olives par exemple, ils ont trouvé de bons résultats pour surtout l'extraction. Donc il y a des zones donc normalement certains hormones qui aident pour la protection du cellules nerveux. Donc vous avez parlé que c'est très intéressant d'essayer la relation entre le figue, surtout donc pas mal de recherches ils ont essayé de faire la relation.
Yes, but I would like to have the interaction between both of them, fig and olive, on disease. C'est-à-dire l'interaction entre, je vais utiliser les figues et les olives en même temps. Quelle est la conséquence sur les maladies, d'accord? L'interaction également. Interaction also, okay?
Merci.
The experimental design give us this results. Okay? Thank you.
Okay, thank you for nice discussion. Okay, now we are going to have Dr. Harok. The topic of his talk is the preparation of herbal mixtures as traditional treatment for dental diseases in Morocco. It's a field survey. Okay, Dr. Harok, please.
Thank you, Mr. Thank you so much. You can see my presentation, please.
Not yet.
Okay. Now it's okay?
Actually, no. I strongly suggest you stop the sharing. Now, okay. Don't do anything else. Now it's okay. Okay, please continue.
Okay, thank you. Thank you. So, hello everybody, good afternoon. I am Hazim Herwak, PhD student in biology from Meknes Faculty of Science in Morocco. I am proud to be here with you today. I would like to thank you very much for this international congress. So my presentation will be about preparation of herbal mixture as traditional treatment for dental disease in Morocco, a field survey. So oral disease have always been a serious and significant problem health, big problem health. Phytotherapy use complex compound included in plant or isolated from them. Modern treatment with herbal medicine consists of the correct use of plant extract. So according to our ethnobotanical survey carried out, plants are used in the form of mixture of several species against oral disease. So for all this reason, we would like to see the relation or the composition of this mixture. To realize this study, we have adopted as methodology for study area, we choose Meknes city. It's one of the 4 imperial cities and 6th largest city by population in the Kingdom. It's located in the north and central of Morocco between 2 seats of mountain, Prairie and the Middle Atlas. For our ethnobotanical survey, we are based on snowball method, and it was conducted among herbalists operating traditional medicine in the city. All collected data was tableaued and presented in the form of graph using Excel 2016. So as a result, we have found like 3 herbalists. This sit, the first one have sit 5 plants in his mixture. The second one 15 plant. The second one have site 14 and the third 11. And for the mixture that have a minimum of plant, it was recorded by 2 herbalists with 3 plant. As you see on the table, all plant name for each mixture. And for our result, we have obtained 42 plants spread over the 24 family, which all are used in the form of mixture. So the most cited one was Oregano compactum named Zatar, with a percentage and cited percentage of 62%, followed by Qronfil and Swag, and we can and others. As I said, we found 42 plants. The most used plant family are Lamiaceae in the first, with 21.67%, followed by Myrtaceae and then Giglantaceae. But the most represented family was also Lamiaceae by 10%, but not, I mean the second one here, it was Myrtaceae. But the most represented, it was Myrtaceae with 3 plants and presented by a 14%. The plants are used generally in the form of their leaves all herbalists they see that this practice are prepared by leaves of plant, followed by roots for other species, or in general aerial parts. In the form of decoction or cooked way and infusion. But there is difference between decoction and infusion, as you know, and some interviewed, they didn't precise if really it's decoction or infusion. They just say that decook the plant and after they take it as solution. So as I say, the galenic form, all herbalists confirm that the preparation it's in the form of solution, fluid sometimes by essential oil or tisane for some Lamiaceae plant especially that we can drink without any toxic effect. It's administered by rinsing way because always we don't we have a problem if we can meet some toxic plant. That's why all herbalists they say that they can tell to their customer or to their client to drink this mixture, this preparation. Also, oral cavity, it's an external organ that we can just rinse or make an external application, for example, for swag, for example. So to conclude, the traditional practices are not always correct. The synergetic or antagonist effect is always a question that arises from a molecule mixture. This result can be a grain analyzed for further in vitro study of their dental effect. And thank you very much for your attention.
Okay. Thank you very much, Dr. Harouk. Actually, I found very similarities of the plant names. You call it zeytun, we call it zeytin in Turkish, as well as I think zahter, we call it zahter in Turkish. So it seems like they have common name in our geographical area as well in Turkey as well as in Morocco. Thank you very much for your presentation.
Thank you so much. Thank you.
Any questions? Okay, if no questions, then we can continue with.
Yes.
Okay, Dr. Mennane, please.
Hazim, please, can
oui.
I ask you in French?
Okay.
Concernant donc l'utilisation de qui sont les plantes à votre avis qu'on peut utiliser pour donc le traitement des maladies dentaires qui sont à votre avis. Donc vous travaillez sur donc, oui.
D'accord. En fait, il y a tout un mélange, une mixture de plantes. Donc, on ne peut pas préciser à 100% sans passer par l'étude in vitro au laboratoire. Parce que durant l'enquête, j'ai trouvé que les enquêtés, l'un à l'autre, donnent des informations qui sont parfois différentes. Donc, ce qu'on peut tirer de cette enquête, c'est par exemple citer quelques plantes qui sont, ou bien recommander quelques plantes qui sont plus citées. Mais on peut rien confirmer sans passer par le laboratoire et l'étude in vitro, parce qu'il y a toujours l'effet antagoniste, ou bien il y aura une synergie entre ces mixtures. Et comme vous savez, lorsqu'on a une mixture de plantes, on a un extrait ou un mélange qui est très complexe. Donc, on ne peut pas, par la suite, dire l'activité elle est due à quelle plante exactement. Donc, c'est tout un processus très compliqué et qu'il faut compléter par des études de toxicologie et tout. Ce que je peux dire, c'est pour valoriser le patrimoine marocain ou les plantes marocaines, il faut bien évidemment éliminer toutes plantes qui ne sont pas marocaines, à savoir soit, par exemple, le qronfel et ainsi de suite. Et concentrer beaucoup plus sur des plantes qui sont endémiques marocaines, essayer de travailler sur les plantes individuellement avant de passer aux mixtures.
Merci. Merci, Hazim.
Merci à vous.
Okay, I think we can continue with our last presenter, Dr. El Mehdi. And the presentation name is the Statistical Inference for Panel Stochastic Frontier Analysis with Dependent Error Terms. Dr. El Mehdi, please.
Okay. You can see my screen?
Yes, it's okay.
Oh, okay, thank you. And I am El Mehdi Rashida from Mohammed First University, Oujda, Morocco. My presentation is about statistical inference for panel stochastic frontier analysis with dependent error terms. It is a mathematical presentation, and I will give the empirical study. So I will talk about panel stochastic frontier approach with time varying efficiency. Why with time varying efficiency? Because we have here panel stochastic, so we have time. In this point, we will talk about the model estimation, how to estimate technical efficiency and make inference on technical efficiency scores. The second part is an empirical study. I will develop the numerical optimization and results of our research. So, at first, I will define the basic idea of the frontier analysis, which is a comparison between the decision-making units firm, for example, in order to know how inputs are used to have outputs using technical efficiency. In frontier analysis, we have two definitions for the technical efficiency. We have Farrell scores and Shepard scores. Farrell scores are between 0 and 1, and Shepard is just the inverse of the Farrell efficiency scores, so they are greater than 1. In our research, we will work with the Farrell efficiency scores, so TE is between 0 and 1. In frontier analysis also, we have two approaches. We have deterministic and stochastic. For the deterministic, the most used is the non-parametric. For the stochastic, the most used is the parametric. For this research, we use the stochastic parametric approach, in which we have the error term is divided in two terms, the noise term V and the inefficiency term U. In the classical way of the stochastic frontier analysis, we consider that U and V, which compose epsilon, which is the error term, are independent in the classical way. In our research, we will consider the dependence between V and U, the noise term and the inefficiency term. So when we have dependence between U and V, the joint density of U and V is not equal to the density of V times the density of U, but we have to multiply by the copula function, which indicates the dependence between U and V. The copula function depends on a parameter T, and I give here two examples for copula function. The product copula is equal to 1. C theta, here F, grid F is the cumulative density, and W is equal to the cumulative density of U, and W2 is equal to the cumulative density of V. If the copula function is equal to 1, is the case of the independence. Here we multiply by 1, so U and V are independent. For our case, we will use the Clayton copula, that is the expression of the Clayton copula, to explain the dependence between U and V. So the model in this case is defined by Y is equal to it's the response is equal to a function of X beta. X is a vector of variables. Beta is a vector of parameters. UIT is a scalar. Epsilon IT is a scalar, and epsilon IT is equal to VIT minus UIT. V is assumed to be normal in frontier analysis, and IT is equal to a time function eta t times UI, and UI is the inefficiency term. It should be greater than 0. Given that we work with panel data, we will use time variant efficiency. For efficiency, we have here 8 model. The 1, 2, 3, and 8 model exists in the literature of the frontier analysis. The novelty of our research also is to handle some time variant efficiency, which take into account the periodicity of the function. So to estimate the model, we should define the log likelihood function. The log likelihood function depends on the density of epsilon, G epsilon. We define the G epsilon by the density, the integral of the joint density of epsilon with UI according UI. So we'll have just epsilon. Epsilon UI is supported by R plus, so the integral is from 0 to plus infinite. The joint density here, epsilon is a vector, UI is a scalar. Why? Because UIT is equal to eta t UI. So UI is scalar, but epsilon IT, if we take epsilon, we have in epsilon I, we have t term according to of the time. So the joint density of them is equal to the joint density of the UI times P density of epsilon I, but we haven't epsilon I. We use VI, and VI is equal to epsilon I minus ui. We have the, we integrate this expression and we will have the G, the density of epsilon I. We define the log likelihood function of our model, and we optimize the log likelihood function using numerical optimization, because no, there aren't our analytical way to have the estimates of theta. Theta here is a vector of parameters which are unknown. We should estimate them. We define, after having estimates, we define the technical efficiency as expectation of exponential minus UIT given epsilon I. It's equal to this expression. And we will make inference on TE using the parametric bootstrap approach algorithm 3 of Simard-Wilson. We will change just step 2 of this bootstrap algorithm. In this algorithm, U and V are drawn independently. Here we will draw U and V dependently using the Clayton-Kopila. These 3 steps are the steps how to draw U and V from Clayton-Kopila, and we repeat the 3 steps n times t times t times to have a pairs of U and V to define the response Y. Our empiric, we have at first validate our computation using simulation, and after that we have applicate our procedure on an empirical study using Moroccan drinking water. For Y, which is the output, we use the production, the water production. For the inputs, we use cells and number of subscribers. For the Kopila, we use the Clayton-Kopila. And we have DMUs is equal the number of DMUs is time period is equal to 7 years. For numerical optimization, we have 3 steps. We fit at first the model, this one, we fit it using the pooled OLS regression. So UIT is equal to 0 and we haven't eta t. Estimates of this step, we will use them to as initial values for the maximum likelihood estimation, considering the independence between U and I, which is the classical way. And step 3, in step 3, we take this estimate as initial value, considering that U and V are dependence in this case. And for this step and this step, we haven't the parameter theta of the Kopila because we have the independence. So in this step, the parameter of Kopila will give some values, so a grade of values. Results. Among the 8 model proposed, we have the best one is the model number 7. So it is a model, a novel, a new model, a novelty. So a new model in the frontier analysis is the best one. Sorry. And for this model, we have parameters, estimate of parameters here, and we have all parameters are significant in the model. So we can estimate technical efficiency. Technical efficiency are here, the estimates. We find that all technical efficiency are covered by the confidence interval, lower and upper bounded, and technical efficiencies for all provinces in their study are low. For several provinces, TE are low, and we find that time effects is significant. Because why? Because we have eta 1 and eta 2 are significant, and all provinces are technically inefficient. We haven't a province with TE is equal to 1. And we have Clayton-Kopila does not approach the product Kopila by a test. So we are right to consider the dependence between U and I in our study. And thanks for your attention. Your remarks and questions are welcome.
Okay, Dr. El-Mehdi, thank you very much. Actually, from high school, I stopped studying mathematics. It was first for me to read this much formula. I wonder, your model, I mean, practically, how can we use? You use the, I think, the water system data there, and then you apply that on, your statistical model. But for instance, could you please give us, like in biological sciences, not like environmental science, but biological science, if you may, how can we use this model actually in biological systems, maybe, if you can?
Yes, this model. So this model is, sorry. I will, so this model is used in different area. So we use it in biosystem, medical area, economic area, mathematical area. So it is used in different area. Just check if we should just check if you can see my my screen?
Yes.
You see my screen?
Yes.
Yeah. Check if yes, if your database is a panel. What is the panel? We have it is a matrix. For the line, we have the I, and for the column, we have a period, T period. Here for my data, we have 7 years, okay? And if the dependence between U and V exists, we should use our model here. I can't. We can use all this computation. If we haven't independence, so the independence between V and U, we can't use this computation because it is new. It's the novelty of our research. It doesn't exist in literature. So if you have independence, there are other procedures to follow. If you have a panel data and you have a dependence between the U and V, U is the newest term and V is the inefficiency, you can use this model in different area.
Okay.
Okay?
Okay. Okay, thank you very much.
Thank you.
I think we reached the end of this session. Thank you very much for all presenters. And I wish you a very healthy, good days and no COVID in future. Okay, please take care of yourselves.
Thank you.